Hello, everyone, and welcome to Anatomy and Physiology 2. This is the first chapter here, Chapter 11, which is on the fundamentals of the nervous system. So, in this chapter, we're going to be doing, like, in Anatomy and Physiology 2, we start out heavy, like I said in the syllabus intro. We're going to be talking about action potentials on the first day here. This is a little bit of a longer lecture, so feel free to pause wherever you like. And don't forget, all these lectures are also available in the files section of Canvas. So if you like to print out the lectures, write notes on the lectures, or look at the lectures as I'm going through it, you have that available to you. So in this chapter here, what is the purpose of the nervous system? We're sensing our environment. So how does that work? What are the cells involved? And what's the differences we're going to talk about? The whole first unit is all on the nervous systems. Nervous system is very complex. So this chapter goes into the different cells that are available, the action potentials, and how a neurotransmitter works and sends its signal. Because one neuron needs to speak to another. So we're going to look at what the nervous system do and how it is organized. And we're going to look at the cells are called neuroglia. And then so those are the cells that are supporting the neurons then. And then we're going to look at how neurons send electrical signals. This is called the action potential when they send the signal long term. So we got to look at how that potential is generated. It's an electrical current that moves through these cells. So we're going to look at graded potentials over a short distance and action potentials over a long distance. Now we're going to look at how neurons communicate with the next neuron. We have to send that signal from one to the next. So that happens at something called the synapse. Now, you did talk about the synapse when you talked about the neuromuscular junction. It's just a slightly different form of a synapse for muscle contraction. But neurotransmitter is released there. In that case, it was acetylcholine. So might refresh you on that a little bit. And we'll talk about how we summate that potential on the next neuron. And is it excitatory or is it inhibitory? Because it could be one or the other. Then we're going to look at some of the specific neurotransmitters and then how neurons work together, different pathways they can take. So here, just starting out with the functions of the nervous system, just obvious stuff here, is the master controlling communicating center of the body. So we have to have a sensory input coming in. It then integrates here in the brain, and then we have a motor output going to a muscle. Sorry, my cat is currently running around and going crazy. I don't know what he's doing. He usually sleeps all day. But anyway, the benefits of recording from home. So if you hear meows in the background, it's him playing with something. I don't know. But anyway, here, nervous system is the master controlling and communicating system of the body, and it communicates via electrical signals and chemical signals. So we have to talk about that process. And it's very, very fast. But sensory input, integration, motor output. So we have to bring in that sense. We have to figure out what it means and then send an output to regulate it. So this is broken up into the central nervous system, which is chapter 12, and then the peripheral nervous system, which is chapter 13. So central nervous system is brain and spinal cord. Peripheral nervous system is everything that comes out of that. So the cranial nerves, the spinal nerves, and the ganglia out here, which are part of the sympathetic nervous system. And then there's the parasympathetic nervous system. So those are all the next couple chapters here that we're going to go into. So a lot of information that we're going to discuss. So then looking at the peripheral nervous system, it has two functionary divisions then. The sensory one, so remember peripheral, anything on the outside. Sensory, bringing stuff in. One is somatic sensory. These ones, somatic, whenever you hear the word somatic for the nervous system, think it's bringing in information from the skin, skeletal muscles, and joints. So the somatic areas to the central nervous system. Visceral, think organs. So these convey impulses from visceral organs to the central nervous system. So visceral is organ, if you remember that from Anatomy and Physiology 1. Both of these are going to the central nervous system. They just get a slightly different name. And remember, these are afferent with an A. And then the motor or efferent divisions, these are transmitting pulses from the central nervous system. So they're exiting the central nervous system, typically exiting the spinal cord. Now, the two divisions of these, somatic ones, are going to skeletal muscles, joints, skin. Then autonomic ones are usually going to visceral organs, small intestines, heart, things like that. So part of the autonomic pathway, sympathetic, parasympathetic pathways. So somatic here, these conduct impulses from the central nervous system to skeletal muscles, and it's voluntary control. You're sending these signals out. Then the autonomic consists of these the visceral motor nerves, and so smooth muscle, cardiac muscle, glands. Talked about all this, and then two subdivisions we'll talk about later. Sympathetic is your fight or flight response. Parasympathetic is your rest and digest response. So they work in opposition and sometimes with each other as well. So this is just the overview of the next couple chapters here. Next chapter is on the central nervous system. Then chapter 13 is peripheral nervous system. So we'll talk about the sensory and motor efferent divisions. And then so somatic nervous system, all that is 13. And then autonomic nervous system is chapter 14, where we talk about the sympathetic and parasympathetic division. So I'll be showing this figure here a few more times as we go through this. And then chapter 15 is the special senses, like vision, hearing, smell, taste, things like that. Okay, now back to the introduction chapter. So we got to talk about the different cell types in nervous tissue histology. Remember, histology is the study of tissues. So we have two main cell types. One are the neurons. Those are the main nerve cells. There are excitable cells that transmit the electrical signals. Now, there are other cells that are in these regions around them. These are called neuroglia, or glial cells, small cells that surround and wrap the delicate neurons. There are four main neuroglia support cells in the central nervous system. So now we're going to be talking about differences between the central nervous system and the peripheral nervous system. Their function is similar, but they have different things that occur around them. One is the type of cells that are around them. Another one is regenerative capability. So here are four main types we're going to talk about. We're going to talk about the astrocytes, the microglial cells, the ependymal cells, and then oligodendrocytes. I'm not going to define them here. I have slides for each one as we go through them, but these is just introducing you to the next little section here. First here are the astrocytes. Astro, think star, astronomy, site cells. So it's astrocyte is a cell that looks like a star, has a whole bunch of things coming off of it. So most abundant, versatile, and highly branched of the glial cells. So down here we see an astrocyte in this image. So these wrap around neurons. So in yellow here is the neuron, and these astrocytes wrap around neurons and also wrap around the capillaries. These form what's called the blood-brain barrier. You will hear me talk about this a lot. Sometimes this is just abbreviated as BBB. So wrapping around the capillaries here controls what comes into and out of the central nervous system. So very, very important. Also, stuff might come into the astrocytes here, be processed in the astrocyte, then sent out in something like neurotransmitter formation and things like that. So here, support brace neurons play roles in the exchange. Most of the things I just talked about. A lot of the functions here are guided by astrocytes. Very, very important part. Next one are the microglial cells. So these ones are interesting because our central nervous system doesn't have the same immune system that the rest of our body has because of that blood-brain barrier. So it has its own macrophages, which are particles that eat things. So these are small ovoid cells with thorny processes that touch and monitor neurons, because there are still neurons that die, there are still debris that need to be taken up. So these can transform through phagocytized microorganisms and neuronal debris as well. So they're the immune responders of the central nervous system. Then ependymal cells, these ones line so in the central nervous system, we have this stuff called CSF. CFF is cerebral spinal fluid. So in the rest of the body, you have extracellular fluid that surrounds the cells and so forth. In the central nervous system is a special fluid called the CSF. So here, actually, these ependymal cells line these fluid-filled cavities, and they have these little cilia on them that beat different directions. The beating of that cilia moves the cerebral spinal fluid. So they form a permeable barrier between the CSF and cavities and tissue fluid bathing the central nervous system cells. So very, very important in moving the fluid around. And then the last ones here are the oligodendrocytes. These are highly branched cells, and then they wrap around neurons in the central nervous system. So oligodendrocytes, central nervous system. I know it says it all over the slide, but I just want to emphasize central nervous system. And what these do is they make myelin sheaths. Myelin is this protein lipid substance that wraps around the neurons. It kind of acts like so when you have an electrical cable, if you would take the rubber off, you would just have the cable in the middle. Think of the cable as the axon, which is this yellow part of the neuron right here. You wrap that rubber cable around it as insulation, and it allows that signal to work properly. And that's what this myelin is like. So these myelins are this fatty protein substance that wraps around these neurons and lets them send their impulses or information faster. So in our body, we have white matter and gray matter. I won't write it all here. We'll talk about it later. So white matter versus gray matter. If you ever hear white matter, it's white because of being myelinated. Gray matter is not myelinated, so you're just seeing the neurons. And we'll talk about that more next chapter when we talk specifically about the central nervous system. So if you're ever doing a dissection on something, white fibers are always neurons when you're looking at it for the peripheral nervous system. But the peripheral nervous system has a different cell. They're not called oligodendrocytes in the peripheral nervous system. They're called Schwann cells, and we'll talk about that later. So here is the later. So two major neuroglia is seen in the peripheral nervous system, not central nervous system. First ones are the satellite cells. These are similar to astrocytes, but they have a different name. So they're forming a supporting role around cell bodies, giving them nutrients and so forth. So this would be the cell body of a neuron here. And then here we have these little myelin nodes. Each one of these is a separate cell. These are similar to oligodendrocytes. However, each one is a cell. And they're called Schwann cells. If we go back to oligodendrocytes, this is one cell wrapping around tons of different axons. Here, one Schwann cell only wraps around one little region of an axon, and that's one big difference. So these are called Schwann cells, and they form these little gaps called nodes of Ranvier, so the signal can jump all the way down and move very, very fast. And these are, because these have Schwann cells, also allows the regenerative capability, so they can connect two regions and bring something back together if something bad happens. Okay, now neurons. So these are the structural units of the nervous system. So we just talked about all the neuroglia. Now we can finally focus on the neurons. And I do have a lab model video going over the model that we have in the laboratory, which is important for you to know for the practical. Knowing the structure of a neuron and different parts of a neuron are very important. So these are large, highly specialized cells that send the impulses. So they bring them in at one end and send them out the other end. So extreme longevity, they should last a person's lifetime. Amitotic, which means they don't divide, and they have a high metabolic rate. They're using lots and lots of ATP. So they have a cell body in one or more processes. So this is general representation of a neuron. So right here we have the cell body with the nucleus in here. That's the nucleolus. And then there's also some chromatophilic substance, which is this stained, rough endoplasmic reticulum out here. Then right here we have mitochondria floating around. And now coming off of this, sending the signals in. So imagine all these pointing inwards here. They're communicating with everything around it. These are called dendrites. So dendrites, think of them as fingers coming out. They're sending the information in. They then summate it. Some of these might be positive. Some of them might be negative. If it reaches what's called the threshold, it will then send the signal down the axon. This point of exit here for this is called the axon hillock. And then it goes down the axon, and right here we have the Schwann cells. And on the model, you can see that we'll talk about the myelin wrappings later and how that works. And then in between, we have the myelin sheath gaps, also known as the nodes of Ranvier. So the impulse is being sent this direction. And here then we have the axon. Down here then we have the axon terminal, and there could be thousands of axon terminals. So the signal gets down here and then sends that signal to the next neuron. So we got to talk about how the signal is sent to the next neuron. So that's just a general representation of a neuron. Make sure you watch the lab video that goes with this as well. So here, just talking about the cell body. So it's where everything is made. Makes the proteins, membranes, chemicals. The rough ER is there too, and also nucleolus for RNA processing, and then some can contain pigments. I'm not going to spend too much on this. Then most neuron cell bodies are located in the central nervous system. Now, just understand some naming differences. Nuclei are in the central nervous system. So when we talked about the central nervous system, next chapter we're going to talk about all these nuclei found in the brain. The suprachiasmatic nucleus is a cluster of nuclei in that region. Then ganglia are outside the central nervous system and the peripheral nervous system. Usually we see ganglia associated with parasympathetic and sympathetic divisions. Okay, so now we got to understand how neurons can move as well, and the differences in naming. In the central nervous system, they're called nerve tracts. So in the brain, different tracts, different association tracts and fibers moving across and so forth. And outside of that, they're called nerves. Now, make sure you don't call a neuron a nerve. A nerve has a whole bunch of neurons in it. I'm not going to draw them all, but imagine it's filled with little neurons. A nerve is then also going both directions. So sending impulses up the afferent division and out the efferent division. And so then the two types of processes are the dendrites and the axon. Right here, just showing different examples of neurons. Neurons come in different shapes and forms. We'll talk about them later. But here I just wanted to show all the different complexity of these dendrites they can have. Most common ones here are the motor neurons, or the multipolar ones. They contain hundreds of these short, tapering, diffusely branched processes. I have a receptor region, and then in that cell body is where they figure out the graded potentials. We'll talk about that later. And then the axon structure here, this is showing a lot of studies done on the nervous system have been done using the giant squid, because it actually has this one single giant axon that you can look at and study. Whereas looking at, a nerve, you're looking at thousands of axons. Here, it can isolate. You can actually see this with the naked eye, one single axon. And all the action potential stuff that we're going to talk about today, a lot of that work was studied using the squid. So some axons can be over 1 meter long, so very, very long. So some sensation you get in your foot when you step on a nail, you're going to, I'll be using this touching a hot pan or stepping on a nail analogy a lot in the next couple chapters, but it sends a signal all the way up to your spinal cord. There's one neuron that goes from that receptor all the way to your spinal cord. And then there's where it needs to send its signal. So it has a long distance it needs to travel. Axons can also have some branches on them. So like here you can see some branches are called axon collaterals. And then at the end they branch profusely and have tons and tons of axon terminals to send that signal. So the axon is then the functional unit. It's sending that signal down, so the impulses transmit them along the axolemma, which is just the cell membrane of the neuron. I probably won't test you specifically on that. Sends it to the axon terminal, where it then secretes neurotransmitters. We'll talk about those more later. Movement can occur in both directions. If it moves away from the cell body, it's called anterograde, and then if it moves toward the cell body, it's called retrograde. So they can go either direction. Mostly things go away from the cell body. But dendrites go toward the cell body to summate the information. And then here, they have very efficient transport mechanisms. So we need a lot of mitochondria at the synapse. We'll talk about why later. They're processed and they divide up in the cell body. We need to send those all the way down the axon. Some axons are very long, to that synapse. So we have to have very efficient transport mechanisms. So these axons actually, so if this is this axolemma, we have these microtubules in here that have these tracks on them that are then have motor proteins carrying mitochondria down to the synapse. Kind of cool how that works. And then here a little bit about the myelin sheath. So the myelin sheath, remember, is a protein lipid substance, and it's acting as the insulation. So when we term something a myelinated fiber versus a non-myelinated fiber, a myelinated fiber has myelin, non-myelinated does not. So these are very slow, these are very fast. Now in the peripheral nervous system, these are Schwann cells. Remember, in the central nervous system, these are the oligodendrocytes. So the plasma membranes have less protein, and so more lipid, so no channels or carriers, so these are good electrical insulators. So now if we look at how these are formed, it's kind of cool. So here we have the Schwann cell, and it begins wrapping around, and it continues pushing the cytoplasm. So these little actin filaments here continuously pushing, and it begins wrapping around. It's also pushing from this side here. So pushing the cytoplasm out as it wraps around more and more and more and more until it forms all these little layers here. And here you can actually see how tight those layers get. So it makes this insulation where nothing can get in and out. So here you can actually, the little dots are the microtubules, and this is a cross-section of a mitochondria as well, which is kind of cool. So that's it being walked down. Now, we have this heavy insulation, and it forms these little gaps, and that's how we send the signal. So when the signal travels down, it jumps from node to node to node to node, rather than having to go through every ion channel all the way down. It'd be very slow if that happens. So these are called nodes of Ranvier or myelin sheath gaps. Either work. I always call them nodes of Ranvier. But technically, they're called myelin sheath gaps too. Either is correct if ever asked on a practical. So, but non-myelinated fibers then are thin fibers not wrapped in myelin, so that signal can go away, and it can only travel short distances as well. So then the central nervous system, again, is different because there's where we have oligodendrocytes. They're not whole cells, and they wrap around multiple neurons. So myelin sheath gap is present, though, but there is no collar of paranuclear cytoplasm, and the thinnest fibers are unmyelinated but covered by long extensions of adjacent neuroglia. So white matter versus gray matter then, told you we'd get here. White matter, regions of myelin. Gray matter, mostly neuron cell bodies and non-myelinated fibers. We'll see that much more when we talk about different regions in the brain. So here now, structural classifications of some neurons. Neurons can be multipolar, bipolar, or unipolar. Just these are what they look like. So multipolar are the most prominent ones. So 3 is easier to show in the images coming up. So here, let's just go to the images. So here's multipolar neuron. Multipolar neuron has a cell body right here with all the dendrites. It's a typical representation of a neuron. Gets a signal coming in, sends the signal down, sends it to the axon terminal. So many processes extend from the cell body and are all dendrites and then a single axon. So these ones are typically abundant as motor fibers. So sending out all the signals moving through your brain, all the ones motor outputs and so forth. Most of your neurons, it's the major neuron type in the central nervous system. We'll talk about Purkinje cells in the cerebellum and also pyramidal cells as well. The next neuron type are called bipolar cells. So these ones aren't as prevalent. Actually, I said motor neurons for this. I did not mean to say motor neurons. So there's a difference in the motor neurons, just to clear this up. Motor neurons have an efferent and afferent. So afferent sensory neurons are multipolar. Now, efferent, I talked about backwards. Let's imagine we're sending the signal in. We just had a sensation, we're sending it in. That travels via a unipolar neuron. So let's write this out here. So afferent means it's going towards the central nervous system. Those are unipolar neurons. They have these things called cell bodies. In the afferent motor division here, sensory division, this is called the DRG, or the dorsal root ganglion. We'll talk about that later. But that is a great example of a unipolar neuron. So right outside, so where, let's say this is our spinal cord right here, where this neuron is coming in, there's a clump right here that comes off. That's called the DRG, and that's where these cell bodies of these sensory fibers are coming in. And now if we're inside the spinal cord, it synapses then through a little interneuron, which is a multipolar neuron typically, and then it sends the information back out. So that then goes out the efferent pathway to the motor unit. So that's what I meant to say there. So there is a difference between afferent and efferent for motor. There's sensory motor, and then also, well, somatic motor, and then actually sending that motor output. So slight difference there. Sorry if I just confused you, but we'll talk about it a little bit more then. Just now, back to the bipolar. Bipolar then has the neuron in the middle. It is different than the unipolar. Unipolar sticks up like a T almost. That's where it kind of gets its name. Whereas the bipolar is kind of like two axons right here. Well, a dendrite and then an axon, and the cell body is in the middle. So you can get all the summation here, and then you go out this way. This one's popular in the special senses. So we'll talk about that. A big one is in vision, where these are prevalent. So here, where we find some of these right here, it's showing the example of the dorsal root ganglion, which I just horribly drew, but it's showing a little better. Here it's showing, for bipolar, showing the olfactory cell and the retinal cell. Retinal is vision. Olfactory is smell. So both of them are prevalent there. Pyramidal cells and the Purkinje cells as well. So major neuron in the central nervous system. Multipolar is the most abundant one. Just make sure you don't screw up that efferent versus efferent, afferent division. And they are different neurons. So now we can have different classifications of neurons. I've thrown these words around a few times now, but we can have sensory, motor, or interneuron. So a sensory neuron senses. A motor neuron is sending out a motor output. So most of these cell bodies are located in the central nervous system for that motor output. Then interneurons are little communications in between. And most of our neurons in our body are these little interneurons. So here, showing a multipolar one. So again, same table diagram here. So here's the interneuron. It kind of starts here. So this is step 1, and this is step 2 for this one. And this is what I drew earlier. So we have a sensation from the skin here, sending an impulse up a unipolar neuron. So this is that little cell body sticking off. That's the dorsal root ganglion. And then it comes in this part of the spinal cord. And you notice the inside of the spinal cord is a different color. That's because this is gray matter in there, and it's white matter on the outside. So inside here, we have these little interneurons, which is a multipolar neuron, then forms a little synapse right here, then sends the signal back out, not a unipolar neuron, and then comes back out to the muscle. So that's just a little feedback mechanism. Imagine this red line coming in right here then, and sending that information through. And then some other ones here are like the retina sending vision to the brain, which we'll talk about later. All right, I'm going to take a little break right here. You feel free to take a break too, and then we'll get into action potentials. I just need to give myself a little voice break. All right, hello everyone, welcome back. Okay, so now we're going to be getting into part B here, which is going over the action potential. This is the little heavier part of the day, if there is one. So we have to talk a lot about ionic movement and charges and electricity, pretty much. That's how neurons work. So neurons have this thing called resting membrane potential, which you'll see me abbreviate as RMP. Unlike most other cells, neurons can rapidly change this resting membrane potential. So neurons are highly excitable. Now, you did talk about resting membrane potential in this stuff in chapter 3, and also for when talking about the cell membrane. And then you also talked about it for the neuromuscular junctions and how that works as well. So this should be a review. So if I go through this fast, make sure you review it and so forth. I'll try not to, but you should have talked about this a lot before. This should be the fourth time you're hearing it if you include the time you heard it in Bio 1. So this is all about the movement of ions and these membrane channels that are in the cell membrane. So there are two main types of ion channels. We have leakage channels. So these ones are just always open. They're always there. And they're non-gated, means there's no gate blocking them. Then also we have gated channels, which means there's a gate. So here would be a leakage channel if I were drawing a cell membrane right here. So, things can go in and out depending on the concentrations. Whereas a gated channel has something blocked where you can't go in and out. It's blocked. So these could be chemically gated, voltage gated, or mechanically gated. So if a chemical binds right here, this gate would open up and now we can go through. That chemical is called a ligand if it binds. Or it could be voltage gated. So here, let's draw this little block coming back here. If this hits a certain voltage, this is really amazing how this works. If this hits minus 50 millivolts inside the cell, this gate actually opens then and then allows, sodium to move in or out. And then mechanically gated, meaning something can move it in causing a mechanical opening. So usually these ones are connected to, let's draw this one a little different for mechanically gated. So typically for this one, there's some cilia, and the movement of the cilia actually causes the channel to open and allows things to move in and out. So mechanical movement of that cilia moves stuff in and out. Super cool how they work. So here, just showing better drawings of some of these. So chemically gated means something binds and opens it up. Voltage gated means the voltage has to be proper for it to work. This is the big one for membrane potential. So if this hits a certain potential, these sodium channels open and then sodium can rush into the cell. So when we talk about this, we talked about the electrochemical gradient. So electrochemical gradient, first the electro part, meaning there's an electrical difference between inside and outside the cell, extracellular versus intracellular. Uh, so when we represent something and say minus 50 millivolts, that's talking about the difference between inside and outside. Now there's also a chemical gradient. So that's the difference in concentration between the ions between outside and inside. So both these two things combine. So let's say we have a high, so when I draw brackets, that means concentration. A high concentration of sodium outside and a low concentration of sodium inside. Based on just the general theory of diffusion, where does that sodium want to go? It wants to balance concentration inside and outside. So sodium wants to move that way. That's this little arrow right here. But now let's look at charge. So we have a high positive charge out here. So positive charge, not really a concentration, is much higher out here. And then positive charge inside is much lower. It's actually negative. And that's what that negative and positive represent here. So now also imagine you were a sodium ion. Let's imagine we're this sodium ion right here. We are a positive charge. So opposite charges attract, and you want to move down your charge gradient. So here, this positive charge really wants to move towards this negative region. So these sodiums are lined up out here, wanting to go inside the cell, but they can't unless a channel is open for them. And then there are also negative ions here, but they typically stay relatively similar. It's the movement of sodium and potassium that are the key here, but potassium is actually the key one involved in building this resting membrane potential. But right here, I just want to talk about the differences between the chemical gradient and the charge gradient. So with the chemical and the electrical, forming the electrochemical gradient, with sodium really, really, really wants to come inside the cell, and that's the key to an action potential. So here, first we have to build the resting membrane potential. So for a particular neuron, it's around minus 70 millivolts. Other cells in the body can be around minus 90 millivolts. So how we measure this, we put this little thing called a voltmeter inside the axon, and then it has a little ground outside, and then it can record the electrical difference between inside that neuron and outside the neuron. And using one of these is how we figured out everything about an action potential and how neurons work. So kind of neat how that works. So this membrane is said to be polarized. Important term here for being polarized. Polarized means it has sides, meaning it has a positive side and a negative side. So compared to zero. So if the charges are coming closer to zero, that's depolarizing. If they're going further away from zero, that's hyperpolarizing. We'll talk about those terms a little bit more later. So here we want to look at differences in ionic composition. I talked about this earlier as well, so I won't dwell on it here. Sodium is higher out here. Chlorine is also higher out here. Potassium, and that kind of balances everything too. Then we also have negative charge proteins, which are the A's in here. Potassium is a higher concentration inside, but there is still a negative charge inside because of these proteins. Okay, so now let's generate this resting membrane potential. Key player here is potassium. We start out with leakage channels. Up here, I'll talk about the sodium-potassium pump later. But first, this is step one. Step one, let's imagine the cell interior is at minus 90 millivolts. So it's there because of potassium leakage channels. So these are always open. There's a couple of them. And remember, if potassium is in here, and this is minus 90 millivolts, it wants to move out to equal its concentration. So it's lost through abundant leakage channels, and that establishes the negative membrane potential. Because potassium concentration is low outside, it wants to move outwards. Not necessarily too low, though, because this is a positive ion. It's kind of happy in here in this negative environment. So not only does it take its chemical concentration into effect, it also takes into its electrical part in effect too. Because look up here. This is called the sodium-potassium pump. You talked about this one before too. So sodium-potassium pump, 3 sodiums out, so 2 potassiums in. So that eventually that will build up a higher concentration of potassiums inside, and a lower concentration of potassiums outside. So potassium wants to go that way. It wants to go down its concentration. I meant to draw a down arrow there. It wants to go out its concentration gradient or down its concentration gradient. However, it has to fight the electrical gradient as well. So it finds a little balance around minus 90 millivolts. Next, we add a sodium leakage channel. So this is weird. Now, there aren't many sodium leakage channels. So there's only a couple, so sodium doesn't rush in. Because you got to imagine, we're pumping 3 sodiums out here. So 3 sodiums out and 2 potassiums in. So a high concentration of sodium outside and a positive charge. That sodium doesn't want to be there. So we throw in 1 sodium leakage channel. Sodium then will come into the cell, and it brings it down to about minus 70 millivolts when that happens. And that's about where a neuron sits. Sodium-potassium pump then continuously, if too much sodium comes in, sodium-potassium pump will push it out if slow enough. So that keeps it all about 70 millivolts. But none of this would happen if it wasn't for potassium starting it with those leakage channels. So now, what can change this potential? We can have graded potentials and action potentials. Graded potentials are when you have little incoming signals, so little influxes of sodium. It all depends on the ion. If you bring a positive ion in, so if inside is negative, you bring a positive ion in, that gets closer to 0. So that is called depolarization. Oops, running out of room here. Now, if you bring a positive ion in, bringing it closer to 0. Now, another thing that can happen, you could bring a negative ion in, such as chlorine. That would move it more negative. So how would I want to draw this? So adding more negative ions to a negative environment would increase the negativity, and that's called hyperpolarization. Run out of room again. Trust me, it says it. It says this word right here, hyperpolarization and depolarization. So that's one thing you could do. Another thing you could do, you could bring positive ions out. So if you remove positive ions from this negative environment, let's imagine there were some sodiums in there, or potassiums, and for some reason potassium's left, that would make it more negative. So you got to think of the charge. Put yourself inside a cell, it's negative. Let's imagine removing positive ions. If it gets more negative, hyperpolarization. If it gets closer to 0, depolarization. That's these terms here. Don't think about it in terms of specific ions. Think of it in terms of just a charge, because we're going to be talking about excitatory and inhibitory things. Excitatory, depolarize. Inhibitory, hyperpolarize. And so it depends on what ion they bring in. So here, if we're looking at the depolarization, so here we're depolarizing. So this means we're becoming less negative. So this could mean positive in or negative out. So here, if positive ions come in, so negative environment, positive ions coming in, we are depolarizing. Negative ions moving out, we could also depolarize. So it depends. And then over here, if we hyperpolarize, that could be positive out or negative in. Both of these effects would make it more negative or hyperpolarization. So a little easier to see it on this graph here. So remember, positive out, positive in, positive out, negative, what am I saying? My words. Positive in, negative out, depolarization. Positive out, negative in, hyperpolarization. And this is just showing the change in, so more negative, more closer to 0, more positive. So to start out with, we'll talk about graded potentials. I have all the words here, but what this means is you get a little impulse and then it goes down. So it travels down, but it will only do anything if it opens a voltage-gated channel. So they're named according to location and function. You have a receptor potential, that's the generator potential, called the generator potential because it's trying to generate an action potential. If we hit an action potential, great, but a lot of times it doesn't. And then postsynaptic potential is then talking about the graded potential that happens onto the next neuron. So here, talking about showing it, little stimulus hits here, So positive ions rush in, cause a little greater potential here, but doesn't necessarily hit action potential and open the sodium channel. So this then just goes down through and then slowly dissipates. So the concentration here dissipates as we move down through. You can also see that represented on this graph right here. But now the important part is what happens if we hit an action potentials. So action potential is an all or none effect. You hit an action potential, voltage gated sodium channels open, boom, we fly. So these do not decay over distance as greater potentials do, and this is also sometimes referred to as a stimulus or an impulse. So if I say stimulus, impulse, all meaning the same thing. So I'm going to talk about it all using this figure here, and in the PowerPoint links, you can also go watch this video. I'm not going to pull it up here, but you can watch this as well. It's a good little representation of all the steps. So let's start out here at part 1. So at part 1, we're sitting at typical resting membrane potential, so RMP. So at resting membrane potential, sorry, dog is getting antsy. We have high concentration sodium outside and high concentration potassium inside. We're sitting at minus 70 millivolts. If we look over here, minus 70 millivolts, and everything is closed. We have, everything's just being monitored and great. But now, we just had a stimulus. So here, some input, let's say it's a chemical. So chemical binds, opens the sodium channel. Sodium rushes in. So now this is a voltage gated channel, and we hit the threshold, which is like minus 50, minus 55 millivolts here. Oops, sorry about that. So minus 50, minus 55 millivolts opens sodium channel. So these blocks move away. Sodium now rushes into the cell. So sodium is going down its concentration gradient and down its electrical gradient. We have a positive ion moving in, so now we shoot up. So this is called depolarization. Depolarization is when it goes up to a very high point, and then we hit a max here at about plus 30 millivolts. At that point, something happens, of course. The sodium channels become inactivated. This little ball and chain feature down here comes in and blocks the channel. It's inactive. You can't move sodium through that channel anymore. It's not the same thing as the activation gates. So nothing can open that channel. No voltage can open that channel at that point. And that is very, very important. We'll talk about why coming up later. So remember that little point right there. So now what happens at plus 30? Another voltage channel opens. Now the potassium channel opens. When the potassium channel opens, so potassium is at a high concentration inside and a low concentration outside. But it's also more positive now inside too. So because of those two conditions, if the potassium channel was the only positive channel open here, potassium will want to rush out. So potassium then rushes out, and now we have a positive out. Remember what a positive out does? We go down and hyperpolarize. Well, in this case right here, it's called repolarization because we're resetting it. But it gets to a point where it actually drops a little bit below minus 70, and then it's called hyperpolarization. So it drops a little lower, and then those channels then close. Leakage channels then come back in. Sodium-potassium pump then plays a role, and we get back to minus 70. And then after these potassium channels close, these sodium channels then reactivate, and this ball and chain moves away. This is important because if this kept opening, the action potential wouldn't travel in one direction. It could travel down the axon, and then that sodium channel hit again, and then it turn around and come back. You want it to go from sodium, voltage-gated sodium channel to sodium channel all the way down through. You don't want it to get to this one and accidentally come back. So if this previous one is inactivated, guaranteed you're going to go this way. But if this one wasn't inactivated and that was already reset, it could, in theory, go backwards. So this makes action potentials unidirectional, traveling one way down a neuron, which is very important. And that's because of the inactivation of the sodium channel and reactivation. This comes in two phases as well. Here, let me erase. I'll leave it here. So here is about where that reactivation happens. So in theory, so there's called the absolute refractory period and the relative refractory period. So about right in here is called the relative refractory period, or well, absolute and then relative refractory period. There's a point in which, yes, you can, in theory, fire another action potential, but it's going to be quite difficult because there you're at, maybe minus 100 millivolts. And then once you get back to resting, it's a lot easier to go from minus 70 to minus 50 to hit another action potential. So the absolute refractory period is absolutely, so that's right here. Once that, well, kind of at the top here, yeah, you're already firing action potential here. It's absolutely, so this little gap right here, you cannot fire another action potential because that sodium channel is closed. And then down here is the relative refractory period, which you can, in theory, fire an action potential, but it's going to be much more difficult. It has to be very high frequency of that initial stimulus. So this is just the general overview of an action potential. All the next slides here do have the words that go with everything I just said. So I will go through and you have access to these slides, but I much prefer to describe it on this figure here. So this brings everything together, and you have this figure for your references as well. So then here, 4 main steps: depolarization, threshold, repolarization. Everything I just went over was on that figure. Remember, action potential is also all or none, so an action potential either happens completely or does not happen at all. It all depends on that sodium channel. And then we move down. So propagation allows the action potential to be transmitted down through it, and it's all about that inactivated sodium channel, and it makes it self-propagating. So one action potential triggers the next sodium channel to open. So this is showing that propagation, and another video that goes along with this that shows it well, showing that inactivated channel. So once it fires here, that initiates the next one, which then initiates the next one, which then initiates the next one. And we don't go this way because these ones are inactivated right there. So that's a no. So now one big thing with action potentials. So what, makes something stronger? What makes a stronger action potential compared to a weaker action potential? It's all about the frequency of action potentials. So a shorter frequency is the distance between action potentials. If you're firing a lot of action potentials in a short order, it has a higher frequency. So this wouldn't be considered as, so this would be a much higher frequency than this comparison right here. So this no stimulus, this is a relatively higher stimulus, but a very strong stimulus fires action potentials very fast. So it's frequency, not amplitude. The amplitude of the action potential does not change. So make sure you never screw that up on something. It's the frequency of the action potential which determines its strength, not the amplitude. So the amplitude never changes on an action potential. So then here for the refractory periods again, I talked about these. So the absolute refractory period is when you can't fire it. The relative refractory period is then you can, but you have to go, you need exceptionally strong stimulus or high frequency. And then here is the little summation of everything. Shows it all pretty well. So make sure you fill in this image too, and you have this all down. You're able to label this one. So absolute refractory period, relative refractory period, depolarization, stimulus, repolarization, hyperpolarization. Make a note here of threshold. You could even draw all over this saying this is when sodium are open, this is when potassium are open, and so forth. So it's really, really important to understand this figure. It will go a long way in helping you in understanding the nervous system. So next part here, how fast do things move? It all depends on 2 things: axon diameter and degree of myelination. So think about, I like to use the straw analogy, and I use the straw analogy when we get to blood vessels as well. So think of drinking a milkshake through a very large straw. It's pretty easy. You don't have to require a lot of force. But then imagine using a little coffee straw to drink that milkshake. You got to use a lot more energy. So diameter, large diameter fibers have less resistance, so have faster conduction. So that's a big one for axon diameter. Well, the same theory comes back for blood vessels and resistance for blood vessels too. And then degree of myelination. How much myelin is wrapped around that axon? More myelination, faster it goes. And not only that, does it have continuous or saltatory conduction? So what's the difference between continuous and saltatory? Continuous is where there's very little or no myelination. So it has to travel all the way down that neuron, sodium channel to sodium channel to sodium channel to sodium channel. Very, very slow. Whereas saltatory, you jump. Saltatory is where you can jump from stimulus is here. It immediately goes right to this next one. So since there are no ion channels in this sheath right here, it goes right to the sheath gap where the ion channels are. So you jump from node to node like I was drawing earlier. So also in non-myelinated axons, conduction is slow. So you can have, it's more like a continuous conduction as we move down through. So it does jump, it's saltatory-like, but not as saltatory as myelinated axons that have these nodes built into them. So this is, I like to bring in all these little clinical homeostasis things here where things aren't working properly, and we think about why they're not working properly based on the physiology we just discussed. So multiple sclerosis, or MS, autoimmune disease, and what it affects is myelin sheaths. So think about what happens if you would destroy those myelin sheaths. Saltatory conduction would not work as well. You would not be getting sensory input or as proper motor output. So this turns myelin into hardened lesions called sclerosis. It's where the autoimmune system attacks the myelin. And so that causes conduction slows and eventually ceases. Now these individuals do go through good periods and bad periods because their body is trying to fix it by throwing more sodium channels in where the myelination might be lacking. So it causes cycles of relapse and remission. Symptoms are visual disturbances, weaknesses, loss of muscular control, speech disturbances, incontinence, everything related to muscle control and things like that, or sensations. Drugs that modify immune system activity, of course, would work, but immune system activity is quite important. May not be able to prevent, but maintaining high blood levels of vitamin D may reduce risk of development. Still studies being done. So then conduction of velocity, you can have different groups of fibers. First one are group A fibers. These are the largest. These are myelinated somatic, sensory, and motor fibers of skin, skeletal muscles, and joints. 300 miles per hour. We'll keep it in United States terms here, or 150 meters per second. Group B fibers are then an intermediate speed, lightly myelinated. So here's where degree of myelination and diameter come into play. About 30 miles per hour. And then group C fibers are the smallest, and they can move about 2 miles per hour, which is still pretty fast. B and C are autonomic nervous system, visceral, motor, and sensory fibers that serve visceral organs. So now let's think about this in terms of other chemical and physical factors as well. So local anesthetics act by blocking voltage-gated sodium channels. If you've ever been to the dentist and they've done extreme work on your mouth, you might have been giving something called lidocaine. Ever wonder what lidocaine does and why it stops that feeling sensation? Lidocaine actually blocks sodium channels. Imagine if we were blocking sodium channels. Would an action potential work? No. But eventually lidocaine is nice because it eventually, once it wears off, meaning it diffuses out, it doesn't have as severe effect and that feeling comes back. So if you block sodium channels, you don't send an action potential. And then cold temperatures or continuous pressure interrupt blood circulation and delivery of oxygen to neurons as well, which could also affect conduction. You need to have the nutrients there. So different things, so opioids, local anesthetics, we won't talk about these other antagonists as well, but things can be excitatory, inhibitory, or block. All right, for the last bit here, we're going to go right in. So if you want to take another break, you can do it now. I'm going to keep powering through. So here, part C is now looking at the synapse. So here, how does that neuron we just talked about communicate to the next neuron? First, we got to get some signals down. So first here is the presynaptic neuron. That's the neuron that is sending the signal. The neuron that's receiving the signal is called the postsynaptic neuron. Most function as both, because then that postsynaptic neuron can become a presynaptic neuron. So there are different definitions we can go through here. One is axodendritic or axosomatic. So how these synapses connect to the next cell. So axodendritic is when it's a dendrite. It's between an axon terminal in one neuron and dendrites of the another. So axodendritic. So here, these ones are most common. So the axon, the synapses are talking to other dendrites. Axosomatic is when it goes on to the cell body or the soma. Less common ones could be axo-axonal. So if these are all excitatory right here, and then this axo-axonal one is inhibitory, it would block the signal. So even if an action potential was hit here, it could get here and block everything that was sent. So that's one big thing that axo-axonal ones do. You can have dendrodendritic, where sometimes a dendrite can talk to another dendrite, and somatodendritic, which is dendrite to soma, but those ones are less common. So then there are two main types of synapses: chemical synapses and electrical synapses. Chemical ones are easier to understand. Not easier to understand, harder to understand. Electrical ones are everything we just talked about. So it's pretty much a direct gap junction between two neurons, and they can send that signal directly on. But first here, I want to talk about chemical synapses. And now, chemical synapses are where we release a neurotransmitter packaged in these little vesicles. They're released, send them out to the synaptic cleft. They bind to the receptors on the other side, and then send that potential down the other one. This is best shown in this image right here, and you have this image, and I want you to draw this one on yourself. Not on yourself, but draw it yourself as well. So here we have the action potential coming down this neuron. When this happens, we have the little mitochondria out here. Mitochondria are releasing all of our ATP because this area requires a lot of energy. Up until this point, sodium comes into the cell. Now a big thing changes. When this action potential gets down here, sodium channels disappear, and they become calcium channels. So right here it's showing these calcium channels. These are voltage-gated calcium channels, so the action potential opens voltage-gated calcium channels. Acts the same as sodium, but calcium, a positive ion, now rushes in. Why is calcium important? Calcium causes these vesicles to go through the release process. So all these vesicles are sitting here at the end waiting to be released, waiting for this calcium. This calcium is a messenger that says, okay, time to be released. These vesicles then fuse. Really neat fusion process how that works. But then they fuse and then release these neurotransmitters. Now, if you remember the neuromuscular junction, this neurotransmitter was acetylcholine. So these neurotransmitters then go out to this area right in here called the synaptic cleft, meaning it's just a very, very close junction between these. And on this postsynaptic neuron, we have these vesicles, not vesicles, receptors. So this neurotransmitter binds to this receptor, and depending on this neurotransmitter and this receptor, you could have different movements in and out. So it varies, it depends. If, again, it's the same thing as before, so if you move a positive neuron in compared to, not a positive neuron, a positive electron in compared to a positive electron out, or ion, not electron, you change that potential. So they can be either excitatory or inhibitory. Now these are just defined whether they're excitatory or inhibitory. If it's excitatory, it's called an EPSP, excitatory postsynaptic potential. If it's inhibitory, it's called an IPSP, or inhibitory postsynaptic potential. So now think about that. If you have, an excitatory coming in here and an inhibitory coming in here, they would balance each other out, and then you get no action potential. The key is, can we make an action potential on that next neuron? So if you have more excitatory coming in or more neurotransmitter being released, you're more than likely to release or initiate an action potential and have enough positive charge coming into that next neuron. So neurotransmitters can be either excitatory or inhibitory, based on a receptor they bind to and how that receptor changes the potential of the postsynaptic neuron. I know I just said a lot of words right there, but trust me, it all fills in as we move through here now. Now, if that neurotransmitter stayed out there, you continuously get this effect. So that neurotransmitter has three different ways it can be removed. One, it can be reuptaken by the original cell, by original presynaptic neuron here, come back up and be reused again. It can also, an enzyme can come out and degrade it for something particular like acetylcholine or something called acetylcholine esterase, which comes out and breaks it up. And this is also a cleft, it's open space. So that neurotransmitter could just diffuse away as well. And remember, we can also have supporting cells out here, astrocytes and satellite cells, which may play a role in neurotransmitters too. Okay, so that was everything. That's all on the next couple slides. So here, how this information moves through is all on these next slides right here. So here, there's also a little synaptic delay I wanted to mention. So it takes time to release that neurotransmitter and go to the next one. I know we haven't talked about an electrical synapse yet, but electrical one, here's presynaptic cell, here's postsynaptic cell. It's immediate. It's just like moving an action potential. Whereas a chemical synapse, these are slower. So there's the action potential in the presynaptic neuron, but we don't get that action potential, like half a millisecond delay, which is pretty fast in terms of milliseconds and things, but it's still a delay. So now the electrical synapses are less common. These ones are joined by gap junctions between the presynaptic and postsynaptic. So it just comes in and sends those positive ions right into the next cell, causing the action potential. Whereas the chemical synapse has to go through that diffusion, has to bind, a more intricate process. Now, these electrical synapses are found in some brain regions responsible for eye movements or hippocampus areas involved in emotions and memory, and then also most abundant in the embryonic nervous tissue as well. So then, looking at how this can come together as EPSPs or IPSPs. So remember, EPSP is excitatory, I is inhibitory. So different ways this could happen. So an excitatory, of course, increases, causes a depolarization. Now it has to hit threshold. This one doesn't hit threshold. An IPSP causes inhibition. Boom. So remember, this is all based on ions. So potassium out, chlorine in would each cause this. So now it all depends on how they summate. So summate is how they add together. So there could be multiple excitatory, multiple inhibitories on the same postsynaptic neuron. So all that matters is what causes an action potential. Now they can come in two types of summations: temporal, time, spatial, over the same area. So temporal is one or more presynaptic neurons in rapid fire order, so high frequency, temporal over a period of time. Whereas a spatial summation is then over the same space. So let's say 3 excitatory hits the same time from multiple neurons. So here showing a few examples. So here, no summation, you have an excitatory coming in, you just get a little boom, another frequency, boom. But temporal, you get 2. So you get first boom, and then second boom, and that hits threshold, and you fire. But now in spatial, you have an excitatory one coming here and an excitatory one coming here at the same time. So boom, boom, fires. But now another thing, you can combine both. You can have the excitatory coming in here and then an inhibitory one coming in here. So here, inhibitory one hit first, then excitatory, and then inhibitory. So they're all competing with each other. So you can have spatial, temporal, all combined into one big happy mess. And then these all can be integrated as well. You can have different modifications. So repeated use of a synapse increases the ability of a presynaptic cell to excite post. So you use it, you learn. This is all about learning and memory. So building more receptors, packaging more neurotransmitter, reuptaking neurotransmitter faster. Whatever increases the chances of causing an action potential in that next cell is learning and memory. A lot of involved in this. We'll talk about this a little more when we get to learning and memory. And then inhibition. So potentiation is sending that signal and remembering that signal. So you have short-term potentiation, which are short-term memories, long-term potentiation, long-term memories. And then inhibiting, so release of excitatory neurotransmitter by one neuron is inhibited by another neuron via that axonal synapse, like I mentioned before. So less neurotransmitters released, leading to smaller EPSPs. Now, a little bit about neurotransmitters here. I'm going to go through a lot of these a little quickly. Just want to show some of the different groups. They're classified by chemical structure and function. So here, acetylcholine is the big one. Remember, acetylcholine we've always talked about up until now as being excitatory. There are some situations that we're going to talk about, such as in the heart, where acetylcholine is actually inhibitory. It all depends on the receptor it binds to. You can also have biogenic amines. So these can be our catecholamines or indolamines. So dopamine, norepinephrine, and epinephrine are made from the amino acid tyrosine. So catecholamines, amines, meaning coming from the amino acids, indolamine, serotonin, histamine, all made from amino acids as well. And these all have a bunch of different effects. So all widely used in the brain, play roles in emotional behaviors, biological clock. Also important, so norepinephrine is important in the autonomic nervous system for fight or flight response. We'll talk about that later. And imbalances can be associated with mental disorders as well. Amino acids then can specifically act as neurotransmitter. So glutamate, aspartate, glycine, GABA, which is gamma-aminobutyric acid. Also some peptides, which are neuropeptides, which are chains of amino acids, substance P, endorphins, gut-brain peptides. We'll be talking about some of these. So somatostatin and cholecystokinin is an important, cholecystokinin is an important hormone in regulating our digestive system, which we'll talk about later as well. So all these are very, very important in this process here. Now, again, you don't have to remember every single one of these. Once we get to the important ones in the different sections, we'll talk about it again. Some can also be purines. So ATP, the energy molecule, is now considered a neurotransmitter, which is kind of neat. So because of adenosine being a potent inhibitor in the brain, caffeine actually blocks. Caffeine has a similar structure to adenosine, so that's why caffeine has similar excitatory mechanisms to it. Remember, purines are those double nitrogenous ring bases. You can also have gases and lipids, so nitric oxide is considered a neurotransmitter. It causes vasodilation, so it could bind with G protein, so G protein link receptors and so forth. So involved in learning, formation of new memories, as well as brain damage in stroke patients and smooth muscle relaxation in the intestine. Also, sulfuric acid acts directly on ion channels to alter function as well, or hydrogen sulfide, not sulfuric acid. And then endocannabinoids, I'm sure we've heard of these before, act at the same receptors as THC. It's also a G protein linked receptor, also considered to be a neurotransmitter. It is lipid based because it's similar to eicosanoids. So may be involved in neuronal development, controlling appetite, and suppressing nausea. So now these effects are grouped into two classifications. So the effects or the actions, mainly excitatory or inhibitory. So effects, excitatory versus inhibitory, and then the actions are indirect or direct. So excitatory or inhibitory, meaning does it cause an EPSP or IPSP? So it all depends on the receptor it binds to. So acetylcholine and norepinephrine bind to at least two receptor types with opposite effects. So acetylcholine is excitatory at neuromuscular junctions, but it's inhibitory in cardiac muscle, all because of the receptors. So here, actions then, they promote rapid responses by altering membrane potential. Indirect action then is where it binds through something. So neurotransmitter here directly opens ion channels for direct. Indirectly, it requires a G protein coupled pathway, which we'll talk about on the next image. You might have talked about them in AMP1. I know I did, but they're pretty much secondary messenger systems. So it's indirectly causing an effect. Neuromodulators then aren't technically neurotransmitters, but they can affect the strength of synaptic transmission. So they could be releasing more enzymes to break up the neurotransmitter. So not necessarily neurotransmitter, but inhibiting the neurotransmitter. They may cause the formation of new receptors. So may influence synthesis, release, degradation, reuptake of neurotransmitter, or sensitivity of the postsynaptic membrane, and maybe released as a paracrine, which is kind of like a hormone that just acts on its neighboring cells. So this is the example of a G protein linked receptor. So it's all linked here in this pathway. So here where the receptor binds, alters this G protein, activates it via GTP, turns on this adenylate cyclase, which takes an adenosine and makes it cyclic, which is called cyclic AMP. So A is the adenosine monophosphate. This cyclic AMP can do multiple things, and depending on the cell. Typically, it causes a calcium release, which then can do different things. So here, that protein then controls the production of second messengers such as cyclic AMP, could be cyclic GMP, don't worry about DAG, or calcium. That second messenger can do tons of different things inside that cell. And the last thing I want to talk about here is neural integration. So I know it's a long chapter, so feel free to take breaks throughout the talk. Neural integration here is when multiple neurons function together for one output or input. So how can one neuron have a feedback on it? Think about walking. How do you walk? You don't think about walking when you walk. It actually is a feedback pathway that comes back through. So let's look at this a little bit. So these different interactions are called your neuronal pools, or your functional groups of neurons. Let's say you have one axon coming down here. Remember, there are thousands of synapses it can make. So this is the discharge zone. Now let's say it's a very, very strong stimulus. So here, all of these have lots of synapses on them, so higher chance of forming an EPSP. But out here, we only have 2 or 3. There's 1 there. So if this was a very, very strong high-frequency stimulus, then the facilitated zone could fire as well, and that would make a stronger input, let you know, and being able to analyze that more. So serial processing then is how the input travels along one pathway to a specific destination. So so one neuron stimulates the next one, or stimulates the next one. So think about it as, a simple pathway. So here, receptor stimulus changes the receptor. Receptor then fires an action potential, comes through the sensory neuron down the afferent pathway, goes through the interneuron, and then goes out the motor neuron down the effector pathway, and then elicits response. So simple one pathway. Our reflexes are our best example of this. We'll talk about many reflexes throughout the next couple chapters here. They're called reflex arcs, but this is just the general features of a reflex arc. We'll break this down more as we go through. Now, parallel processing, where input travels along several pathways, so different parts of circuitry deal simultaneously with the information, and this is important for higher level mental functioning. Think about doing a math problem. Think about, you have your orders of operations, and those orders of operations might come from different directions in the brain. Or a sense smell may remind one of odor and any associated experiences. I always use the example of smelling a wood shop. When I smell a wood shop, I can picture the wood shop I had as a child growing up, working in the wood shop with my dad by just smelling the sawdust. You can form the picture, elicit that memory from the associated experiences with that particular memory. So here then, circuits are these patterns that form, and there are four types of circuits: diverging, convergent, reverberating, and parallel after discharge. The words might be confusing, but easy to understand once I show them on the images here. So a diverging circuit, think about one input, many outputs. So it diverges as it works its way down, so many outputs. So a single neuron in the brain can activate 100 more motor neurons. So think about just going like this. I need to activate a bunch of motor neurons to go like this, but it was one neuron coming out to that. A second type here is the converging circuit. It's many inputs for one output. So different sensory stimuli can elicit the same memory. So here, if I see a wood shop, if I smell a wood shop, I don't know if I can taste a wood shop, or if I think about a wood shop, I can then output that wood shop memory with my dad. So an example of a converging circuit. And then here, a reverberating circuit. This one's important for walking. So the signal travels through a chain of neurons, but then there's one neuron that sends the signal back to the initial. So if this one hits threshold, and then this one hits threshold, it sends it back, and then this one hits again. So it keeps reverberating then. So it keeps firing without having to use that input anymore. So breathing, sleep-wake cycle, and repetitive motor activities such as walking. So very important. So reverberating, think about it as like bouncing back and forth. And then here, a parallel after discharge circuit. So we have the input coming in, and it breaks apart into multiple parts, and then comes back to one. So one input, one output. So slightly different than these other ones. So parallel after discharge. So here, running parallel after discharge is kind of what it means. So think this one's an example of doing a math problem. So you have the order of operations. This one might be multiplication. This one might be subtraction. This one might be addition. Then you bring it all together for one output, which should be the answer. So mathematical calculations is the best example here. And then to end here, I just want to talk a little bit about development. So the central nervous system, we all form from this little neural tube that wraps around right here, forming this little neural crest. If this doesn't fold properly, it's known as spina bifida, where the actual spinal cord can pop out if that neural tube doesn't form correctly. And now, how do all these neurons grow and find their way? I just mentioned this here because this is really cool. This is called a growth cone. So these kind of like sniff out where they're going, and they try to find synapses and receptors. There are these little nerve growth factors during embryologic development where they're all trying to find all these connections. You actually lose a lot, but so the body has a redundancy where it makes so many where you form enough. So once an axon finds a target, it must then find the right place to form the synapse. So kind of cool. These are called growth cones. And then last thing here, about 2/3 of neurons do die before birth because of that. So if axons do not form a synapse with their target, they are triggered to undergo apoptosis, which is the cell death. Cell death isn't a bad thing. You don't want to have those neurons there if they didn't find a target. Many other cells also undergo apoptosis during development, so it's a normal process. There's nothing wrong with this. Neurons are amitotic after birth. However, there are a few special neuronal populations that do continue to divide, such as olfactory and ones in the hippocampus. But we'll talk about those when we get to those special systems. But that is all I have for the first chapter here. Hopefully, you don't go too crazy watching this. I do have the laboratory video of the neuron that does go with this chapter, so make sure you watch that as well. And don't forget, all these files are available on the Canvas. And if you do have questions, definitely reach out to me and let me know. But that's all I have for today. Hope you all have a great day, and bye-bye.